Yes, good evening to you. Anyone is there? My voice is audible to you. Oh, thank you, thank you. Good evening, thank you. And yes, happy Diwali to you also. How are you? Everything is fine on your side? I'm just sharing the link. Okay. Okay, okay. Why you are tired today? May I allow to know why you are tired? Yes. How are you all? Everything is fine on your side? Okay, semester is ending, so in a rush to submit everything like assignments, projects, lots of things are there, and of course you have your maybe your final exams also within the next week or in the next month maybe. So yes, I think you are quite busy, but I wish you all the best to all of you for your great success in the future, for your goals which are there in your life. Best wishes are there from my side, and definitely I know you all will do the best in your own life. Yes, I just want to say you one more thing that tomorrow is our last session. If something is left, then I will share the PPT of it also. But I want to tell you all that we are not, this is a 10-day journey, but we all are connected, okay? So please remain in touch with me. If you need any type of help, any type of queries there, please let me know. I will definitely help you all. So there is no worry of any type of hesitation or something. You can call me, you can message me at any time. I am always there for all of you. So no issues at all regarding anything. If you want any type of help or something, you can let me know also. Yes, we have Payal here. Payal, good evening. How are you, Payal? Rashmi, today is only the last session of this 10-day lecture series because they said me to cover it in 4 days. I think from Monday, but I started from Tuesday basically. So some things which are not covered, I will definitely cover today. And then if something is left, then I will share the PPT of it so that we will be able to get everything regarding the lecture series. Moreover, some students are also, saying that they have their exams, they have their presentations also. So that is why we are already very late. Otherwise, this session will be completed in maybe 10th of November, maybe I don't know. So we get late little bit also. Yes, Geetika, good evening. How are you? Yes, it is the last day of this lecture series only, not about your internship, not. It's the last day for your lecture series only because tomorrow is Diwali and we are all will be busy in the festival which we have. So I will try to take some extra class so that we will able to complete whole thing. So if something is left, then I will share the PPT or we will discuss it at any time. Not on this lecture series, but at somewhere else also. Everything becomes so hodgepodge basically. Um, you know, otherwise the things are planned so well that I want to let you know about something, some softwares and everything. But the plan is not worked for this lecture series. It happens sometimes. But I don't want the loss of any one of you. You all are my students, and I want that you will get maximum benefit from this lecture series. So I will share the PPTs with all of you. If you will not be able to get anything, you can call me. You can call me at any time, and I will definitely discuss and everything with all of you. Okay. मुझे चार उधर घर के कर देगा So I will try to, I don't want that you will feel something sad or something like that. So this is from my side that I will let you know if there is any doubt or anything is there in the presentation. I will share every presentation with all of you. Just go through the presentations, just go through the information. If you are not able to get anything, then please let me know. We will definitely cover it either personally or at any level, but we will try to cover it. So that is from my side only, so that you will get the maximum benefit of this lecture series and don't feel sad about anything. So happy Diwali, Peshni. Bolina is here. Good evening. How are you? Priyashri is good evening. Priyashri, how are you? So, like yesterday, we have done the software, and I told you some homework. I told you to select some sequences, the FASTA sequences, then we have to run those FASTA sequences on the Mega software. So I think some of you must have done this part. You have selected some sequences, some FASTA sequences. If you have selected some sequences, then please let me know. If you have select some FASTA sequences, please let me know, because this is the homework which I gave you yesterday. So I think some of you will definitely done it. If you have done those sequences, then we will take the session. After completing today's lecture, we will take the session on Google Meet or Zoom also, so that I will be able to tell you about everything regarding the Mega software. So first let me know whether you selected the sequences or not. If Pratik is here, he has done some homework. He has selected some sequences from the proteins of Salmonella, E. coli, Mycobacterium and Bacillus. So I hope some of you definitely search some sequences, some FASTA sequences, so that we will be able to run it on the Mega software. If you have done it, then please let me know. Indrani, are you here? Today you are here. Present, please let me know if you are here. Because I want to take the Mega software on the Google Meet. But if you have done something, then please let me know. Some students are there who already done this software. So if any one of you done this software before also, please let me know. Any one of you. If any one of you has done this software before also, please let me know. And how many of you has done the homework? I told you to find some FASTA sequences of humans, of chimpanzees, rat and mouse. How many of you have done this homework? Please let me know. Please let me know. I'm just waiting for your replies. Please let me know. Please let me know, any one of you. Have you done the homework or not? Otherwise, I will just complete the topic. If you have done it, then please let me know. Otherwise, it's okay. Just let me know only. Okay, I will repeat it again. I will try to repeat it again. I thought I will cover the today's topic. We have some small topics left, which is genome mapping, gene mapping, genome analysis. Small topics are left. Then after it, we will do it on Google Meet or Zoom app. But let me know how many of you have already done the homework, or how many students are interested in doing the software. Just it's my request to all of you that whether you done it or not done it, it's okay, but at least reply to me, all of you, please. So it's my request to all of you, whether you done it or not done it, please reply to me. Gopal has done some homework. Very good, Payal. So is there any PhD scholar there who has done this software before also? Indrani, are you present today? No issues, Priya. Priyashree, we will discuss it again. No issues at all. So, any of the PhD scholars, no issues, no issues. We will discuss it today also. But let me know any of the PhD scholar is present who told me that they have done this software. Indrani, are you present? Or I think one more are there. Are you present today? Any one of you, please let me know if you are present. If you are present, any one of you, please let me know. I think they are not present, maybe. So I will cover the small topics which are there, which left on the previous days. One is our genome analysis, and one is gene mapping. And then after it we have some small topic which is about protein prediction, as you already done protein classification, protein structure, and we have done proteomics also, but protein prediction is the one topic which is left. So I will try to cover it today. If I will not be able to cover it today, then I will share the ppt with all of you, because the ppt is ready. So then you have to go through it by your own self and let me know if you are not able to get anything. Okay, getting my point? If you are not able to get anything, then you have to let me know. We will discuss it on Google Meet or on some another platform. The students which will not present today or something which is left, we will definitely discuss it at some other time. But just let me know. Yes, yes. And one more thing I want to tell you all, that it's okay if you want to do the review paper, till it's okay. If you don't want to do it, I don't force you. I don't want to force any of you because it don't look good or nice. So it's your own wish. If you want to write the paper, then you are able to submit it to me by 25th of November. So today is 13th of November. You have 10 days, or you can say 12 days more along with you. So you can submit it to me by 25th of November, so that we will be able to publish the review paper in the end of November. It will be your achievement, it will become your achievement in the year of 2020. So just try to write a review paper, and your paper will be published in a good journal. So when you will go for any type of interview or any type of you can say exam or something, at that time this paper will be, you know, beneficial for all of you. So it's my suggestion, it's not my force, but it's my suggestion to all of you that if you want to write the review paper, I am there for helping you all. The last date of submission is 25th of November. After it, we want to publish it within the month of November or December. So let me know if any one of you wants to write the review paper. Then we will do it together. I will help you definitely. Okay. So I think that's all for the, you can say, yes, yes, Daveen, I will. Today is your last day, so I have done with your lecture series. So I was little bit busy about with this lecture series also. So now I will be free. I will try to check it today at the night, or I will check it tomorrow. So you just message me by tomorrow 12 p.m. Then I will let you know. Otherwise, I will answer. I will reply you on your mail also. So no issues. I will share everything and I will help you how to write a review paper. And yes, one more thing I want to tell you all those which are there in their bachelors, which are the newcomers. Okay, for the newcomers, we have our another, group, which is bioinformatics review group na. Basically it is for the newcomers, but the use of the group, it becomes for some another purpose, because it becomes very different group, not according to as we wanted, as we wished. So I request all of you, or I suggest all of you, to write some review paper. It will be helpful for your future projects also. Getting my point? So the topic, the our group, which is bioinformatics review paper group, in which you all are newcomers or you are bachelors. So I will suggest all of you to write some review papers. It will be beneficial for all of you in your future perspectives, in your future, you can say, targets or goals. So please write the review paper. It will be helpful for all of you, and it is not as difficult as you may be thinking, or something is like that. It is easy to write. Yes, yes, Pratik, you have to send me the abstract first, along with the title of your, along with the topic, or you can say title of your paper. Then I will mail you if there are some corrections or something is missing. After it, you have to send me your full review paper. Getting my point? Valina, uh, no, it's not compulsory to submit the full paper first of all, and I don't know how to submit. Well, if you want to submit me the abstract and get the certificate, I don't know. I am not aware about this thing. For it, you have to concern the Edufabrica team. I really don't know. I'm sorry for this thing. But yes, I want to tell you that I am not forcing any of you to write the review paper. So you are free from my side. Rest is depend upon the team. Okay, so you are free from my side. I am not forcing any of you, but other things are depend upon the Edufabrica team. So you have to concern with once, because I don't know about this thing. So today we will do. Yesterday we have done phylogeny. Drug designing, we already done it. Proteomics is done. Screen technology is there. Sequence alignments we have done on the very first day. So today I will start with genome analysis and gene mapping. After it, we have gene identification and prediction, and protein structure prediction. If we will able to discuss it today, it's okay. Otherwise, these are simple presentations. You have to go through by them by your own self. Okay. So I will start with the genome analysis and gene mapping, which is very small topics which are there in your syllabus. After it, all of your syllabus is complete. You are very free to check your syllabus. If something is left, then we will discuss it later, or I will share the PPT with all of you. Okay. So now we will start with genome analysis basically. Yeah. So it is the very basic topic. It is the very simple topic. All of you must know about this. These topics we have to cover on the very first 1 or 2 days. But because of some issues, we are not able to cover these topics. And then I just moved to the proteomics part because the other teacher told all you about the protein structure. So I start from the proteomics part. But this thing is left, so I will try to cover it today. So you all know that what is genome? Genome is the entire complement of a genetic material of an organism, virus or an organelle or haploid set of chromosomes in a eukaryotic organism. So basically genome is the entire complement of genetic material of an organism. So whole genome is the complete genome set of an organism. Whole genome, whole is nothing, you can see only pronounce it as genome sequencing also, is a laboratory process where complete DNA sequence of organism genome at a single time. So when we are doing the sequencing, sometimes what happens, we are just focusing on the single gene like CO1 gene, CO2 gene. Sometimes we are focused on the mitochondrial genes only. But when we are discussing about the genome, then we are doing the sequencing of whole genome at a single time. So, whole genome sequencing plus bioinformatics. So, the term sequence analysis implies subjecting a DNA or peptide sequence to sequence alignment, sequence databases, repeated sequence searches, or other bioinformatic methods on a computer. Sequence analysis in bioinformatics is an automated computer-based examination of characteristic fragments, example of DNA strand. So when the sequence or when we are able to add the bioinformatics part, then we are able to automatically or computer-based examination, we are able to do the computer-based examination by bioinformatics softwares which are there. With the help of all those bioinformatics softwares, we are able to do sequence alignment, we are able to do sequence database. We have so much of database, so we are able to generate the results on the basis of bioinformatics softwares. So, genomic analysis is the identification, measurement or comparison of genomic features such as DNA sequence. So, we are able to do the comparison of the DNA sequence. Suppose we have thousands of DNA sequence of a single gene. Suppose we want to do the sequencing of a single gene like any gene which is there, mitochondrial gene, suppose CO1 gene. So we are able to do the comparison of those sequences with the help of bioinformatics softwares. So genomic analysis is the identification, measurement and comparison of genomic features such as DNA sequences, structural variation, gene expression or regulatory and functional elements at a genomic scale. Methods for genomic analysis typically require high throughput sequencing or microarray hybridization and bioinformatics. So when we want to do genomic studies, when we want to do comparison or identification of these genomic features, this DNA sequence, structural variation or gene expression, we have to use the bioinformatics software. For genomic analysis, we have to need the bioinformatics software. So this is the history of sequencing. Sequencing, when we will be able to do the sequencing after doing the PCR. Suppose by amplification of a gene, you are able to amplify a particular segment of a DNA or you are able to amplify the particular gene. After it, you have to do the sequencing of it. So it was developed by Maximum and Gilbert in 1976 to 1977. Since this method was technically complex and use of extensive hazardous chemicals and fallen out of flavor, so Sanger developed the chain termination method in 1977. So we have the DNA sequencing which is there. Only be used for fairly short strands, and longer sequences must be subdivided into smaller segments. So basically what's happening here, we have the sequencing, we have two types of, like we have chain termination method and we have dideoxy method. So this is the history of genome sequencing, that bacteriophage, which is FX174, was the first genome to be sequenced in 1935. So you are able to know that from 1935 we are able to do the sequencing of the genes. So, bacteriophage was first genome to be sequenced with 11 genes of 5368 base pairs. So, when we are doing the genomic sequencing, we are able to do the whole genes which are there. So, they have done with 11 genes and 5368 base pairs. This is a viral genome smaller than T phages and are polyhedral. This way done by Norman and Baker by staining method with the use of Sanger method of shotgun sequencing. So the history of genome sequencing is quite old. Then we have Haemophilus influenza was the first bacterial genome to be sequenced in 1995. This is a gram-negative bacteria with 1.8 million base pairs. So why we are doing the whole genome sequencing, or why there is a need of doing this type of genome sequencing or genome analysis? Why there is a need of study the whole set of genes? Why? Why we want to do to sequence every single gene? Because we will be able to know information about coding and non-coding part of the organism. So if we will do the whole genome sequencing, then we will be able to know the functioning of every, you can say every base pair or every sequence or every gene. So we will be able to come to know which part is coding and which part is non-coding. We will be able to know exons, introns, which one is helpful for which one is there for disease detection. So when we are doing the whole genome sequences, we are able to know the coding and non-coding part of the organism. We are able to find out important pathways which are there. We are able to find out the evolutionary study and species comparison. Yes. So for this species comparison, when we want to do the species comparison, we have to do the sequencing and we have to run the bioinformatics software. Like yesterday I already told you, suppose we have 11 species. We have 11 species. We are able to know that how much similar they are and how much dissimilarity they are carrying. And even I show you that one which is very near to India, which is the species from China, they have very closely placed in a phylogenetic tree, and one species which we have taken from Slovenia, that species was quite far away from the Indian species. So we are able to know the evolutionary study and the species comparison. It is more effective personalized medicine when we want to do to detect any medicine or to detect any disease, when we want to make some drugs. For that we must know the genome, basically the whole genome sequences is needed at that time. Identification of important secondary metabolites which are there in case of plants. Then we have to know about disease susceptibility prediction based on gene sequence variation. So these are the steps of genome analysis. When we have to do the genome sequencing, when we have to do the, by using bioinformatics softwares, what we have to start with? We have to start with genome sequence assembly, identify repetitive sequences, gene prediction is also there, train a model for each genome, genome annotation, metabolic pathways and regulation. Why there is a metabolic pathways and regulations? To find the missing genes, protein 2D gel electrophoresis. So these all are the steps of genomic analysis when we are doing the whole genome analysis. So Like in the last step, evolutionary modeling to analyze chromosomal arrangement, duplication predictions can be made. So this is the timeline of large scale genome analysis, from when and where it starts, and like it starts from 1984 and up to 2001, how much we are able to do the things, like on mouse how much we are able to do, on humans the whole, this is a whole scale analysis, genomic analysis with a period of time. Gene prediction and recognition is also in your syllabus. Gene prediction in eukaryotes, even in prokaryotes, we have two types of gene prediction also. So it is easy, I have a separate ppt for this also, I will share with all of you. It is easy for prokaryotes, one gene one protein, more difficult for eukaryotes. So when we are working on the prokaryotes, it is very easy, not very easy, but it is little bit easy to do the work on prokaryotes as compared to eukaryotes, because eukaryotes are multicellular animals, and they have many proteins, and it is very difficult when we want to do it on humans. Gene prediction is very difficult in case of humans. Short exons separated by non-coding long introns. Gene recognition is by sequence alignment. So when we are using the software, when we are putting our sequences, there are approximately 100 or maybe 1000 sequences, then we do the alignment by cluster W. After it, we are able to know that how many sequences are similar and how many dissimilarity is there. Human genome data, you must have done it in your previous classes. It is definitely the topic in your syllabus also, and lots of you must have done it in your exam also. So human genome data, you all aware about it. Difficulties are there. Small genes are very hard to identify. Some genes are rarely expressed and do not have normal coding usage patterns, thus hard to detect. Human genome data, this is the whole scenario of the human genome data. It was started in early 1990s, and it was predicted for 2003, that means around 5 years, but the cost is so much for this whole human genome data sequencing which we have done. Then we have next generation sequencing. This is the recently developed technique. This is a recently a number of faster, cheaper sequencing methods have been developed. So in 2006, however, this next generation sequencing, we are not using it yet on the, you can say, on the research level for the students, but yes, the technique is new, and lots of researchers or scientists, they are doing work on this next generation sequencing also. So in next generation sequencing, we have array-based sequencing, sequence full genome of an organism in a few days at a very low cost. So if you want to do the sequencing by the Sanger sequencing, it is very much highly costed. You can say the cost is high in case of Sanger sequencing. For single sequence, it takes, even from students want to amplify a single gene and they want to do the sequencing of that single gene, they have to pay around 1000 for that single sequence. So that is very costly for the students also. So but this generation sequences, it's little bit low in cost and produced high throughput data in very short reads. So these are the instruments which are used in this next generation sequencing. So towards next generation sequencing instruments, these are the instruments which are used. We will not discuss about these instruments, but yes, these instruments are there. Like Illumina sequencing chemistry, solid sequencing is there. So basically these all are the steps for the sequencing of the whole genome basically. So you just go it by your own self. These are the methods of sequencing only. Like P454 FLX, Solid, Illumina, Iron Torrent, they are low cost, and we are able to do the sequencing within very short period of time. So they have some benefits also. that faster diagnosis is there, improve outcome is there, and of course it is, its cost is low also. So all these things are there. So genome analysis is basically what a gene is sequenced to predict its function or to manipulate its activity or expression. So availability of genome sequences, if we have whole genome sequences, we will able to do the sequence of all the genomes of an organism, so that we important genes, we will able to know the important genes which are there, and we are also able to know the disease which are there by a particular set of genes. So these are the challenges which are there: gene prediction, phylogenetic history, presence of repetitive sequences, genome rearrangements. These all are the challenges which are there in the genome analysis. So you have already done. We have already done about this. The cytolized DNA, heterochromatin, new heterochromatin. You already know about these things. It's again same. After it, we have gene mapping. So we have genome analysis, and after it we have gene mapping. Now what is basically gene mapping? So gene mapping describes the methods used to identify the locus of a gene. You all know that, na? Mapping, map. So it describes the method used to identify the locus of a gene. Oh, sorry. And the distance between the genes. So the distance between the genes and identify the locus of that particular gene. The sense of all genome mapping is to place a collection of molecular markers onto their respective positions on the genome. Molecular markers come in all forms. Genes can be viewed as one special type of genetic marker in the construction of genome maps and mapped the same way as any other marker. So genetic mapping is basically what? Genetic mapping is based on the use of genetic techniques to construct maps showing the position of genes. So basically mapping is done, gene mapping is there to know the position of the genes, the distance between the genes, the locus of the gene, and other sequence features on a genome. So we have genetic mapping and we have physical mapping also. Physical mapping uses molecular biology techniques to examine DNA molecules directly in order to construct maps showing the position of sequence features including the genes. So like this is the example which is there in case of Drosophila. This is a genetic map and we have to work on the recombinant frequencies. All these things you have done in your bachelor's or in, even in plus 2, these things are there. So like this is the eye color which is there, like vermilion eye, yellow body, white eye, miniature wings, rudimentary wings. So we are able to do the distance between the pair of genes. Okay, so this is there in case of genome mapping. We are able to do the, not do the, we are able to know the distance between the pair of genes. So physical mapping includes restriction mapping, fluorescent in situ hybridization, sequence tagged sites which is known as STS mapping. So what happens in a restriction mapping? Which locates the relative positions on a DNA molecule of the recognition sequence from the enzymes which are restriction endonuclease. And when we are using fluorescent in situ, we are using the probes. So we are using the fluorescent probes basically, which gives us a color. Then sequence tagged sites, which is STS mapping, in which the position of short sequences are mapped by PCR and or hybridization analysis of the genome fragments. This is known as STS mapping. So this is physical mapping. Physical maps can be generated by aligning the restriction map of a specific piece of cloned genomic DNA. These maps are extremely useful for the purpose of map based gene cloning. So each vertical bar represents a echo or one side. So this is a physical map. So physical mapping is there, restriction mapping is there, chromosome mapping is there, and DNA sequencing is there. There is some difference between the genetic map and physical map also, that there is a very little recombination takes place in heterochromatin, a small distance in the genetic map. So genetic map basically consists of the whole, you can say whole chromosome basically. So very little recombination takes place in heterochromatin. A genetic distance, a small distance in the genetic map corresponds to a large distance on the chromosome. So like from this to this, the euchromatin distance is, for a physical map, the euchromatin distance is 54.5, and heterochromatin distance is also there, and again the euchromatin distance is there. So physical map divides it into three, but in genetic map we have to cover the whole distance. Now what are the uses of gene mapping? Why there is a need of to know the distance between the genes, to identify the genes where they are present? Why? Basically we are doing it for the purpose of to know the diseases, or we will be able to know the genes which are responsible for that disease. Like there are some heritable diseases are there, cancer is there, some diseases are there which are heritable in nature. To know about those diseases, we must know about the genes which are involved. This is a human genome project. You all know about it. It is human genome project. It is an international scientific research project with the goal of determining the sequence of chemical base pairs which make up human DNA, and of identifying and mapping all of the genes of the human genome from both a physical and a functional standpoint. The Human Genome Project originally aimed to map the nucleotides contained in a human haploid reference genome. The genome of any given individual is unique. Mapping the human genome involves sequencing multiple variations of a gene. There are some limitations of that Human Genome Project also, but our main focus will remain on gene mapping. So gene mapping is basically determining the location of elements within a genome with respect to identifiable landmarks. So we are able to know the location, or you can say we are able to know, identify the genes which are present at which end of the chromosomes basically. So we have genetic mapping, we have physical mapping, we have comparative mapping. So, in genetic mapping, linear description of DNA markers or genes on a given chromosome with closely placed markers being inherited together more often. What is physical mapping basically? It is a physical location on the chromosome relating more towards exact positioning of the gene elements. So, we have genetic mapping and we have physical mapping also. Linkage is there, which is there in your genetics part. Bolzan is the one who is able to tell us about linkage. These all things are there in the genetics part, like these back cross, F2 intercross, pedigree analysis, linkage, IIT mapping. All you have done it in your previous classes. Recombination distance is there. Then mapping with pedigree, in which we are able to find the disease, that how the any type of genetic information is passed from the mother to the son or from the father to the daughter. Then these are some testing for linkage and types of linkage. We will not do these things. Restriction mapping is there. Yes. So this is the processing of the sequence data, that from how we will, this is the processing which is there in the bioinformatics part. So by using the BLAST software, we have not done the BLAST yesterday, but we will do it today. So by using the BLAST software, we are able to collect the data. So by using the BLAST software, we are able to get the local and public data. This database, this database, which consists of raw sequences, processed sequences, BLAST results and summarized results. From this database, we are able to get the results. Then there are some gene mapping methods also. This is a history, like Morgan is the one who is able to tell us about the eye color which is there in the fruit fly, and then Wilson is there who is tell us about the sex-linked genes which are responsible for the color blindness and for the hemophilia in human beings to be located on the X chromosome, similar to the many X-linked factors being described by the Morgan group in flies. Types of gene mapping, genetic mapping and physical mapping, it is same again. Then these are some biomarkers which are there, like RFLP, which is restriction fragment length polymorphism, in which we are using the enzyme which is restriction endonuclease, and we are able to break the DNA, you can see. So this is the RFLP, and then we have sudden hybridization, in which we are using the gel. We are doing it, there is sudden blotting for the separation of the protein parts. SSLP is there. These all are the techniques which are there in ambiguity. DNA chip technology is there. Then genetic mapping in bacteria is by transformation, conjugation and transduction. This is again physical mapping, which includes FISH, which is fluorescent in situ hybridization, and it also includes STS mapping. So in physical mapping we have restriction mapping. Sorry. So in physical mapping we have restriction mapping, we have FISH and we have STS mapping. All this is fluorescent in situ hybridization, which enables the position of a marker on a chromosome or extended DNA molecule to be directly visualized. In optical mapping, the marker is a restriction site and is visualized as a gap in an extended DNA fiber. In FISH, the marker is a DNA sequence that is visualized by hybridization with a probe. So yes, there is a probe which will give us the color and we are able to detect it. So there is addition of the probe, and after it we are able to detect it by the color. STS is there. This is the importance of gene mapping, and why there is a need of gene mapping, or what's the importance of this gene mapping. Gene map is the anatomy of the human genome. It helps in the analysis of heterogeneity and segregation of human genetic diseases. It helps to develop methods for gene therapy. It provides clinically useful information about the linkage. This is an important book, the book from the Brown, C.A. Brown book name is Genome. If you want to read this book, it will be quite useful for all of you. Now this is genomic data analysis, in which we are using the computational challenges, in which we are using the software for assembly, for alignment, for analysis, and at the end we want to do the storage of the data. So from double helix to sequencing, like we first of all we are able to know the structure of DNA in 1953, and after in 1970 Sanger is the one who tell us about the sequencing, and then in 1990 we are able to do the Human Genome Project. The goal of this project is to identify the sequences of all the genes of the human genome, which will be expected to be more than 1 lakh. So human genome project is quite big project till now. So it started in 1990 and its expected ending time is 2003. Now, what are the results which are there after this human genome project? Locate and sequence and understand the function of the at least 1 lakh human genes. So, we want to do it on 1 lakh genes. This is again the history of this human genome project, what happened in 1998, then in 1999. In 2000, in a press conference at the White House, the US President Bill Clinton and UK Prime Minister T. Blair, together with both C. Vendor and F. Collins, announced the first draft of human genome sequence. So, they are able to crack the, or you can say they are able to do the sequence or human genome project, which is there. But it is very costly. This is again new generation sequencing, which we already discussed before. These are the computer-based softwares which are used like Lumina, Solid, Iron Torrent, Roche 454, Pacific Bioscience, Oxford Nanopore, Moleculo. These all are the computational softwares. Polymorphism is there. So I will share this PPT. You just go through it. Then this is genomic data banks like GenBank is there, in which we are able to do our, in which we are able to store our data, or from where we are able to collect the data for our own purpose, for our own analysis. We are able to collect the data from that softwares. So this is all about genome analysis and gene mapping. These things you have done in your previous classes also. So I am not focusing that much on this presentation. I will share with you all of you. If you will get any problem, then please let me know. Okay. If you get any problem regarding it, then please let me know. Otherwise, I will share it with all of you. Yes. So is it clear to all of you? If there is any query, please let me know. Otherwise, we will move forward. Please let me know. Is it okay? Shall we move further? Yes. So one more presentation is there, which is regarding the this gene identification and prediction, and another is regarding protein structure and prediction. I will do only one, and the rest I will share with you. You have to go and let me know if you are not able to get anything. I just want to focus mainly on the software part right now. This is the topic, which is the gene identification and prediction, which is there. But before starting it, I want to know, have you download the Mega software on your laptops or not? Please let me know, so that we will do that part first. Please let me know. This is the mega software which we already done yesterday also. So you have to open it like that way on your own laptops. So when it gets open, you have to add your pasta sequences. So I already told you yesterday also, like we want to do edit and delete alignment because we are working on the DNA. So I just asked you yesterday also, just go on GenBank and you have from here you have to search the pasta sequences. Okay, suppose I want to do on, like I did on, suppose we want to do in case of mosquito. Okay, mosquito, and we will take some insects like. So I'm just writing CO1 gene in Aedes mosquito. So from here we have to select the pasta sequences. Okay, so yes, if you will able to see that from we are able to get 12,000 results. Okay, so from here I am just taking the pasta sequences because I want to show you how to run the phylogenetic tree. I am just doing copy, and I am just pasting it on suppose notepad. So I am just pasting it here, paste. Then again I am just doing it for 4-5 sequences. Then I will go with this third sequence. Even you are able to download the pasta sequences of all these 20 also. If you want to do, you can do it. So I am doing copy and I am doing paste here. Let us check whether we are do it for whole sequences or not. This is the option of download here somewhere. The option of download is there somewhere from where you are able to download the whole sequences at a one time. Yes. Suppose we want to do the pasta only. So from here I will be able to get the FASTA sequences directly. So from here I am able to download all. Okay. So I have to do copy and paste only. Copy and paste. Copy. So I'm just selecting as many sequences as I want. Copy. This is the thing which I told all of you to do. But I think none of you have done it, or one or two may have done it. So you have to copy and you have to do paste. So this is the sequences from mosquito. Then after it I will take the sequences from butterflies. Suppose I will take the sequences from butterflies also. So I'm just writing here, just going back, and I will write here in butterfly. Suppose in butterfly, or in case of any other insect is of your wish, in butterflies. So are you getting it or not? Are you able to understand it? Please let me know if you are able to understand it. So yes, I just want to do it on only the FASTA sequences goes. So these are the sequences which we get, the FASTA sequences. So suppose I want to download the FASTA sequences, but I have to select how many sequences I want to download. 1, 2, 3, 4, 5. So 6 sequences I want to download. So from here I will download it. 6 sequences I want to download. So these sequences I will able to get. For these six sequences directly come in the mega software, or you have to copy paste in the notepad also. Are you getting my point? So from here I just download the sequences which I selected, and just I am able to get it in the mega software. So I have my another sequences which is of mosquitoes. So I am just doing copy, and I am also adding these sequences here. So these are my mosquito sequences. So these are my mosquito sequences. After it, suppose I want to do the CO1 in which insect you want to do, in which insect? Suppose in cockroach. Suppose in cockroach you want to do. Are you getting it or not? Please reply. Any one of you, if you are listening to me, please reply. So from here I am just doing the pasta, and then I will select only, suppose I will select only 4 sequences. Then I have to do send to file, the 4 items save file. So these will directly come in under my this case. I'm pasting here. So I have my these sequences which are there. So I have my mosquito sequences, I have cockroach sequences, here is the butterfly sequences. Just I have what I have to do now. I have to do the alignment by cluster W. So I have to do the alignment by cluster W. So I will do it right now. So okay. So if you do the alignment, what you will get? Let me show you. Yes. These stars represent the sequence similarity in all the species. Okay. These stars represent the sequence similarity. After it, I will do the phylogenetic analysis. Just go there and we have to do the phylogeny. Suppose we are using neighbor joining tree. Suppose these are the trees which are there. Like here you will be able to get the similarity. So the mosquito species, if you are able to see Aedes aegypti, these species are placed together. Aedes rusicus, they are placed together. But these species of cockroaches, they are placed together. They are quite far away from the species of Aedes mosquito. Getting my point? So by using these phylogenetic softwares, we are able to do the species identification. We are able to know the divergence which is there in case of species. So from here we are able to get that they have some common origin, but there is so much difference that cockroach species they are present here and they are quite far away from the Aedes species. Are you able to get it or not? There are so many options here in which you can draw it in any pattern, in any format also. You can do it compressed also, rooted also, in straight in this form also. You can do it in any form. In circle also, so you are able to see it in circle also, like we discussed yesterday. It is up to you how you want to check it. So are you able to get it or not? Please let me know. Any one of you, please reply if you are able to listen to me. So any one of you, please reply if you are able to listen to me. Any one of you. Please let me know if you are able to get it. Are you able to listen to me? Please reply. Priyashni, please reply if you are able to listen to me. Okay, okay, then please reply. Please reply if you are able to understand it. Please reply if you are able to get it. So that is basically all for this software part. If you if you are able to do it by yourself, please let me know. Ma'am, is the cluster? Yes, yes. In this mega software, when you go there, if you have in your laptop also, so here you have to align. So you can align by cluster W, you can align by muscle also. So if you want to do by this muscle is also alignment app, which is multiple sequence app, multiple sequence alignment. So from here also you are able to do the alignment of the sequences. There are the stars, so you can do by cluster W also. And if you are working on you can say protein parts, then you can do it by cluster W codons. So, if you are working on protein part, then you can do it by cluster W codons. So you are able to get the this part. So you can do alignment by cluster W. Even you can do here, you can change these sequences, these nucleotide base pairs into protein part also. Let me show you. Like protein sequences, 1 minute. Yes, yes. So I can change these nucleotide base pair, which is adenine, thymine, cytosine and guanine. We can change it into protein sequences also. So now you have to do the 1 minute alignment. By using this, you are able to, you are able to know the presence of stop codons if there are any. And even you can do reverse complement also. So yes, so this is the protein sequences or the codons which we get. Okay, so now they are codons which are now present. So this is the protein data which we get. First we are working on the DNA sequences, but now we are working on the protein sequences. And even you are able to do the, you are able to find the codons also. Let me show you. We are also able to find the codons. Now we will do the alignment by muscle. Compute. So this is the alignment which is by the software, and these are the results. And if we want to do the phylogenetic analysis, we can do it by this also. So are you getting it? Yes, yes. Manish is here. How are Manish? How are you? Happy Diwali to you. Do you know about the mega software, Manish? Are you do you know about the this mega software? Please let me know. All of you, are you able to understand little bit of this software or not? Please let me know. All of you, Priyashri, are you able to get it or not? So these are the translated protein sequences. So we are also able to work on these protein sequences as well. And we can work it on DNA sequences. So from here we are able to get the phylogenetic tree. Even we are able to know about some another things which are there. The conserved sites, the variable sites which are there. So are you able to get it little bit, any one of you? Please reply if you are able to listen to me, Priyasri. If you are able to listen to me, please reply, any one of you. My voice is audible to you? Okay, thank you. Thank you, Pasna. Thank you, PhD. So, Manish, I am asking you, are you able to listen to me? Please reply if you are able to. Manish Mehta, are you able to listen to me? Please reply if you are. So you can do the comparison by any sequences which are there in GenBank. Even you want to, you can do the comparison, like even I told you yesterday, to collect some sequences from humans, that is homo sapiens, from rat, from mice and from chimpanzees, and then we will do the comparison. That is why I told you to select some sequences or some faster sequences from the GenBank, so that by doing the comparison we are able to know the evolutionary history which is there in these mammals, you can say, or you can say in humans and in chimpanzees. So I told you yesterday also to select some sequences which are there in the gen bank. Okay, so I suggest to all of you to select some sequences and do this by your own self in your own mega software which is there in your laptop. So you have to select the sequences from the gen bank. Yes, very good, Priya. Very good, Priya Shri. If you tried with three sequences, I want more from you, and it will be so nice of you if you will be able to share your, the screenshot of something. If you will be able to make, if you will be able to make some phylogenetic trees, all of you, it's my suggestion to all of you. Please make the phylogenetic tree like this tree. Please make the phylogenetic tree and share it with me in the our group. Okay, so the screenshot of this phylogenetic tree, all of you please. Okay, okay, Manish, okay, okay dear, okay. So all of you have to make this phylogenetic tree and share with me in the WhatsApp group. All of you, it's my suggestion to all of you. If you will be not able to get anything, then you have to let me know. I will definitely help. Okay, so yes, if like Manish, you have your seniors who are operating the software, you can also take their helps also. They will help you in understanding the software much better. So it's my suggestion to all of you to just go here and You can select any sequence. Okay, like Homo sapiens, I want to select on the Homo sapiens mitochondria. Suppose that means I want to do work on the mitochondria DNA. So there is a nuclear DNA also. There are some nuclear proteins and there are some mitochondrial. So I have so many of options, like this chromosome number 5. So I have to select it only. So from here, from summary, I will go to FASTA. So I will able to get the FASTA sequences only. And then from here, I have to go to I have to select how many sequences I want. Suppose I want 5 sequences from humans. So I have to, either I have to copy paste it from here. Look how much big this mitochondrial sequence is. That is why I just want to write some small one like CO1. Sorry, CO1 only, because the sequence is too big. So by this you have to do the comparison. Like I am just working on the CO1 gene. Suppose I am just want to do it on the CO1 gene. So you can use any gene, CO1, CO2, cytochrome B, 16S is there, NADH is there. You use any of the gene sequence, because we want to do it, we don't want to do the whole mitochondria, rather we are working on a small gene sequence. So like we have this much of number, just I want to select 5 only. So I love, let us see, check, these are very small sequences, and then from here I have to do the file, and I have to create the file. So this is a file which gets created, and this directly come under the mega. So these 5 sequences come under the mega. So all these are the sequences from Homo sapiens. Similarly, 5 sequences from chimpanzees, 5 sequences from rhesus monkey, 5 sequence from rats, which is Rattus rattus. So you can write here Rattus rattus also. Rattus rattus is our rat which is there in our homes. Rattus rattus CO1. So I can also select the 5 sequences from Rattus rattus. So I will select the 5 sequences from Rattus rattus, and then I have to paste it on a notepad or copy it on a notepad, and then I have to paste it here. So I will paste it here. Like these are the mosquito species which get pasted. So I have to paste it here. So I have mosquito species and suppose human species. So what I will do, I will do the phylogenetic, I will do a cluster first. Okay, then I will make the phylogenetic tree, and I will tell all of you to make the phylogenetic trees and send me in the WhatsApp group. So I will do the phylogenetic analysis. Yes. So from here, yeah, we will go there and I will construct the neighbor joining tree. Now let us see how much resemblance is there. Yes. So this is the tree which we get. So Homo sapiens, Homo sapiens, Homo sapiens, they are in the separate group, and Aedes species are in separate group. This species, which is Aedes aegypti, is little bit closer to this Homo sapiens. So maybe this species, they may be from both from the same side. So this is the comparison. This type of a phylogenetic tree you have to make, and you have to send me in the group. Okay, like Anopheles, Rosicus, they are 81% similar. Aedes vexicans, it is 55% similar with these Rosicus. Then we have Anopheles, which is there. So you have to make such type of phylogenetic tree, and then you have to share it with me also. So are you able to get my point or not? Please let me know. Any one of you. let me know, any one of you are you able to get it or not. We have to do the comparison by using humans, that is Homo sapiens, then chimpanzees, rhesus monkey, and ratus. So you have to do the comparison by using 4 5 animals or organisms, you can say. So please, anyone of you, please, if you are able, then please let me know. So I want this phylogenetic tree from all of you also. Please make it by your own self also. Even this cluster W software, Priya, it's also available on Google also. So if you want to do it on Google, so plus you have to write here cluster W. Okay. So from online, like this is online. So you have to add your sequences here online. Let me show you. So I have to paste the sequences here. So I will do the copy and then I have to do paste. Okay, but then you have to do all the setting like this, these line, there is a gap now, so you have to do all this setting if you want to do it on online software. And then we want to do the DNA sequences, so instead of protein I click on the DNA. Okay, so then you have to do the execute, you have to execute it. Now this is the error because I have not corrected the sequences which I have to correct first, these. So you have to correct it first. But these are so many, that is why I am not correcting it. So but just by doing this you will be able to do the, you have to do it by your own self, that is by manually, otherwise it will show error. Okay. Okay, I will select only two sequences. Wait, I will do with two only. So I will take these two sequences. Okay, and paste. Okay, so you have to, there should not be any gap here in the sequences. You have to do it by your own self. Otherwise, this will not recognize, the software will not recognize it. Okay. Yes, there is one more gap. Okay, and that gap is there. So you have to, I hope now there is no gap. You can also do it online also. Yes. So just execute the results. Yes. So these are the stars are there which are showing the similarity between the two sequences. That A is there, the base pairs, the DNA sequences which are similar in the two sequences, the base pairs which are similar. So you are even able to know that one is 670 base pair long sequence and other is 536 base pair long sequence, and you can then use any of these methods to execute the result. Suppose we will do fast A. So we are able to get the results online also. So these softwares are available online also. So these are the result files which we have to download, then we will be able to see it. Like, let me check anyone. So these are the software which are there, like bootstrap method is there, fastful is there. You can use any of these. Even you can use cluster W using the protein sequences also. Cluster is there. Faster sequences when we want to work on faster sequences. So these are some softwares for protein sequences, like this is PRRN, and we have MAFFT. So we have to submit it and we will be able to get the results. Okay. Okay, good, Priya. Very good, very good. Just try to do it by once by your own. Definitely, it will help you in your future perspectives also. So it's my suggestion to all of you to done it by your own, and you will get, it will be useful for you in your future perspectives or in your future. If you want to do some type of research work, these softwares are quite useful to all of you. So is it, are you getting it? Are you not feeling bored today? Everything is fine. That's all from my side, which I want to tell you. Even one more thing is there, which is blast. In blast, what basically we do, let me tell you. This is basic local alignment search tool. When we work on the nucleotide sequences or DNA sequences, then we have to do it on blast M. Let it open, then I will tell you. It's not opening. Let me close this. Yes, this is nucleotide blast search. So this is blast, which is basic local alignment search tool. And that is not working fast. So here you have to add your sequences basically. I will show you. The network is little bit slow. I don't know why it's not opening. So here also this is a for the sequence comparison. Blast is there for, it's for basically, let me show you. It's not opening. And even we have PubMed where we will be able to get the literature or data also. So if it gets open, I will let you know. So it's basic local alignment search tool. It's not opening. So if it gets open, I will let you know. Yes, Sanjita, there are lots of other things which are there in Mega software. We are able to do, we are able to do, we are able to compare the pairwise distance between the species. If we want to compare the distance between species, we will be able to do, like, so this is the comparison, or this is the distance between the species, the pairwise distance. And one more thing which is important for all of you is the, yes, this thing. We are able to know how many conserved and how many variable sites are there. So we are able to know the sites which are conserved Like from 939 sequences, 177 sequences are conserved, and 714 sequences are variable, and 383 sequences are parsimony informative size. So these are some another things which are there, and this is the protein, or you can say the amino acid code which we get from the mega software. Lots of things are there. When you will do it by your own self, you are able to get so many points. You will be able to know everything which is there in the mega software. So basically, why I told you phylogeny, because I want to let you know about the evolutionary history, or the evolutionary rate which is there, or the species which are there, which are how many similar or how many diverse they are, how many sequences, how they are placed together, how many similarity is there, and how much dissimilarity is there. So it depends on the type of work which you are doing. Even we have different types of trees, phylogenetic trees. We can use maximum likelihood tree, we can use neighbor joining tree, we can use maximum evolution tree. Each one has their own features. I am not tell you about these all, the features of these all. Yes, we talk about the distance based methods and character based methods, but all these have their own features. Like some students, they are using UPGMA method, but now this method is quite old. Nowadays, neighbor joining and maximum likelihood tree is more common. So when you keep exploring the things, you will able to get so much new things, and which will be depends upon the type of work you are doing. Like the type of work which is there in your research project, according to it you will modify your, or you can use the software according to your work which is there. Yes, okay, good, Pratik, very good. So lots of things are there. It's my suggestion to all of you. It depends upon your research work. That means how, which type of research work your guide will provide you, according to which you have to generate the data. There is so much of data, there is so much of information, but it is up to you, or it is up to your guide who will help you to do which thing. You are using the software for which purpose. So basically, mega software we are using for the evolutionary studies. Okay, for to know the phylogenetic studies, the similarity between the sequences, or you can say how many sequence divergence is there. So for all these purposes, we are using the software. I hope it will be clear to all of you. Yes, like this here, you can put any of your FASTA sequences in BLAST. Suppose I am putting here this sequence of mosquitoes. Oh, sorry, I am just go paste. So I am just copying it. Copy. And then I will paste here, paste. Now we have to do BLAST. Now just wait for the results. So it's just taking a little bit of time. Just let it open. So all of you, it's my suggestion to all of you to go to the software by your own self. Keep exploring the things. You will get definitely new things. So yes, so this is a sequence which I paste on NCBI, which is KC. The sequence name is KC855598. Na? So this is KC85598, which is the name of the species, or the name of the organism is Aedes rusicus. So I have done the BLAST 10. When we are using the nucleotide sequences, then we have to do BLAST 10. So from this sequence, we are able to found the sequence which are similar to my own sequence. So I am able to get so many sequences which are similar to my this sequence, which is Aedes rusicus. So these are the sequences. Like there is 100% similarity. This is the my sequence KC855981. It is showing the similarity but with other species, like another is here Aedes cinereus. Aedes cinereus. So I'm not selecting anything. So this is the maximum score, and we will be able to know how much similarity is there, how much Curie cover is there, and how much E value is there. So your E value should be 0. It should be 0 when we want to do the, or when we want to find the sequence very similar to my own sequence. So if you will move down, you will able to get there is little bit dissimilarity. They are not 100% similar now. Like here it is 100% similar, here it is 99.8% similar, but later on the similarity gets decreased. So you can select any sequences here, any one sequences you can do, and then you have to do the alignment. So by selecting the sequences, just select the sequence. I have to do the alignment. So I have selected 9 sequences, and I am just doing the pairwise alignment. So I will download. So from here, the sequences, I will able to get the sequences, and I will able to know how much similarity is there. When I am opening the download file, what I will able to see. These FASTA sequences, the 9 sequences which I downloaded, they came here, and also here I am able to know the similarity which is between Curie and between the subject. So this is another feature of DNA sequences or Genman sequences. You have to just keep exploring the things. You will able to get so much of things. Just you have to keep exploring. We just touched the phylogenetic software part. I hope it will be little helpful for all of you. Anjita, I will suggest, because this phylogenetic software, this mega software is not in your syllabus, I just let all of you know about this on my personal behalf, because I think if I have something to share, then I definitely have to share with all of you. If you will see your syllabus, this mega software is not in your syllabus, and you students are just focused on your syllabus, that this drug part is there in our syllabus, we have to cover it. That is why so many days of us are wasted. Otherwise, I want to do these things very thoroughly and very deeply. Even on our second day, we just start the mega software, and even on our very first day, I told all of you to download the software. But because of some misunderstanding, or maybe because of any reason, our project, our lecture series gets delayed and delayed. But no issues at all. I will suggest all of you to just go through by your own self by first. Just try it by your own self. If you are not able to get it, I am there. I will definitely help you. Even not on this, then I will help you on phone call or by using some Google Meet or something like that. But you have to do some efforts also. Okay? Then I am there with all of you. No issues at all. We will discuss it thoroughly also. And Anjita, in which class basically you are pursuing your masters? Please let me know. We will discuss it thoroughly, no issues at all. Just go by it once by your own self. If you are not able to get it, then please let me know. So in which class you are, Anjita? Okay, she is pursuing MS Biotech. Yes, it's quite useful for you also. Not for only you, but for the whole class. Just go through it by your own self first. If you are not able to get it, then please let me know. I will definitely help all of you. So just make the phylogenetic tree and share with me in the group. I will check it and then I will guide you further. If you have some point or if you want to share something, all of you are most welcome. Please let me know. Please let me know if you have something to share. Please let me know. Any one of you, if you want to share something, anything, please let me know. You all are welcome. Okay, then I will share the presentation with all of you. With any one of you, I think yesterday I shared with any one of you, and then you will just share it in the group. Okay. So if you have any another doubt, please let me know, and please, this is our last lecture series or last day of the lecture series. Please let me know if you have any query. No, no, no. It's not the only that we will go with faster sequences, not at all. But yes, when we are doing it for the first time, then faster sequences are a little bit easy to take. Easy to copy paste these faster sequences on a notepad, and it's very easy to copy all these things and do it on and then to paste it. Like we have another sequences also which we can use. Let me show you. So it depends upon the type of work which you are doing. It depends upon the guide which is giving you the work. According to it, you can modify your work. No issues at all. Yes. So these are the CO1 sequences. So these are like faster sequences. We also have another sequences in which you are instead of faster, you are able to directly in which you are able to directly paste the sequences. So like this you are able to these are the sequences from insect species, like there are 20 species from the insect, so you are able to directly paste here. Our main motive is to add this nitrogenous base pair, which is adenine, thymine, cytosine, guanine. So you can use it. There is another in GenBank also. Instead of FASTA, you can download it as a APN file also. So you can use it any type of downloading format, but the only thing is that you have to paste it here in the Mega, and Mega only accepted the nitrogenous base pair, which is adenine, thymine, cytosine, and guanine. So you can you can download any type of the softwares or files, but at the end you have to in the Mega you have to add it as a adenine, thymine, cytosine, and guanine only. Getting my point? So you can add any sequences, as much as sequences you want to add, it's up to you, and then you are able to get the results also. Like these are the trees which I have made before, so you are able to do the comparison. Like there are so many of species, I came to know that chlora ligata, they and chlora ligata same species are 91% similar, and they are showing the resemblance with chlora rusiana. Similarly, Carbola insoucia and Carbola insoucia are 100% similar. Similarly, Nizara viridula, Nazara viridula are similar, but now there is some variation also. Like in between one species, which is Nizara viridula, Nizara anteata comes. Are you able to get it? I don't know if you are able to see it. I don't have any marker also. So, if you are able to see here that in between Nizara viridula, Nizara anteata comes. Now, what does it implicate? How this another species come in between the same species? How? So, there is some either this species is not Nizara antenita, this is Nizara viridula, or how these species come in between. So there are some species which are known as cryptic species, which are morphologically similar, that means from the outside, sorry, which are morphologically different, that is from the outside they are different, but at the gene level they are similar. So the reason may be that Nizara viridula and Nizara antenata, maybe they are the single species, they are not a different species, they may be the single species. So these are the things which are there in the phylogenetic tree. Lots of things are there. So I hope you all will get it, and you will definitely work on it. My best wishes are there with all of you. Are you able to get it? Please let me know. Okay, okay, good, good. You will able to understand it. Just go through it once. All of your doubts will be clear, definitely. That's 100% sure. Just go through it by once by your own self. If then anything is not clear to you, then please let me know. I will be there and I am there along with all of you. Just go through it and let me know if there is any problem. And another thing is, by 25th of November you have to submit me your review paper. This is from my side. Not particularly, Anjita, it's not particularly any website or something. These in bioinformatics you have so many branches: chemistry, physics, biology, math, statics, everything is there. So there is not any, even not a single software is there. Now if I am talking about this mega software, there are so much software similar to this software which are working the same thing. So I will not suggest, I am not able to suggest a single website or something like that for, for bioinformatics only. No, it is a huge field, lots of new things are there. So I just help you that you just go with one or two things first. After you keep exploring the things, keep exploring the softwares basically, not the things but the softwares. Keep exploring the softwares, because there are thousands of softwares which are present online, and sometimes they are working on the same thing, sometimes they are doing some different aspects also. So there is not a particular website here. Yes, if you want some type of literature help, then I will help you. I will share the content with all of you. If you are talking about this thing, then please let me know. If you are talking about some content, some material, I will provide you, no issues at all. But there is not any particular website, there is not there. Yes, if you want any type of material or any type of PDF or PPTs, please let me know, I will help you. Yes, Priya, this is our last lecture today. Yes, you just let me know on which topic you want the literature. You want on the software parts, on the software or on this phylogenetic software. Please, I will share the PPT today also in the group. So just keep in touch with me. Yes, Priya, it's last session for today. Today is our last session. because tomorrow is Diwali and it's a functional day for all of us. And best wishes from my side. Happy Diwali to all of you in advance. Take care of yourself also. Yes, Sangeeta, I will try to share about everything, the knowledge which I have regarding the softwares. I will share with all of you. But just keep in touch with me also. So if you have any another thing to say, please let me know. Actually, Priya, they are the thing which is told me by team is that I have to teach for the 4 days, which is Monday, Tuesday, Wednesday, and Thursday. I think we have started our classes from Tuesday onwards. So I have completed 4 days, which is 8 hours. But if something is left, please let me know. We will cover it definitely. You are my students, and if some topic are left, please let me know. We will cover it. No issues at all. Only 7 days has been covered. Oh, yes, yes. It everything happens, because of the disturbances which are there in between. Because we have to, I think we have to cover it by 10th of November maybe, or 8th or 9th of November maybe the last day. I think so. I don't know thoroughly or completely, but no issues at all. We will take extra class also, but let me know which type of thing you want to understand. I have no problem in it, but let me know the topic which you want to cover, then I will help you regarding it. Okay? Yes, yes, I understand your concern also. I definitely with you, because you also pay for the session. You are also, everyone wants to get the best from the services, no issues at all. But from my side, I almost touched everything which is there. Yes, yes, Sanjita, please don't be sad. I don't want my students to be sad. Please don't be sad. Even we are together, it's not that we are not together. We will have to do the review part also. So I am in touch with all of you. Even my students which have done the lecture series on October or in September, they are still in touch with me. We are writing the review papers. We have done with two three review papers till now. So no issues at all. Why you should not be feel sad. I'm there with all of you, and if something you want to learn regarding bioinformatics, you are most welcome from my side. Just let me know. I will definitely help you. Yes, yes, I will share the PPTs. I have so many PPTs to share with all of you. I will share it today also. And if you are so much interested in the software parts, then I will share the PPT regarding the softwares also. But yes, one thing is that the softwares, like Manish had said, some here his some seniors are doing the software, the mega software. It depends upon the guide, your teacher who gives you the homework or who gives you the project, and he told you about the softwares also, that on which software you have to work. But I will share the softwares, no issues at all. I will share the software knowledge with all of you. But software part is not there in the syllabus, Anjita. If you go through the syllabus, there is not any software part which is written in the syllabus. So there is only the basic terms which are there, which is proteomics, which is protein structure, gene mapping, genome analysis, and your drug designing, drug development. First, I want to go thoroughly with the software part, but students are saying, ma'am, this part is there, we want to do drug designing and something. That is why the series becomes so much disturbed. You all aware of it, I know. So please don't be sad. I am there with all of you. Yes, you have to do a little bit effort by your own self, then I will definitely help you if you are not getting any point. Okay. Okay, dear. Okay, no issues, Anjita. Let me know which type of software you want to do more. I will help you. Okay, no, because we have to test the theory also, because there are some bachelor students also. I understand, Priya, I understand, but the format of this lecture series is totally different. I have some bachelor students also who called me who want to know about the basics, because sometimes what happens when we are doing bioinformatics, we already know that the students will know about transcriptome, about proteomics, about genomes, but what happens basically they even don't know about they didn't know about genomics or proteomics. So that is why it becomes a little bit difficult for us to do it. Okay, let me know which type of software you want to learn. Please let me know, don't be shy. I will help you. Please don't be shy, or there should not be any issue. Let me know which type of software you want to, you want to learn. Please let me know. Priya and Anjita, please. Which type of software means which type of theory part you have covered in your classes, in your school or in colleges. Which type of theory parts which you have you covered, or which type of software you want to learn about. Let me know, please. Any one of you. Ma'am, do you have experience in Pymol? Okay, Pymol, Autodoc, and protein docking. But these things are not written in the syllabus also. Yes, yes, yes. Yes, Jerry, I understand your point of view also. Even Anjita, these things which are you asking about PayMol, about Autodoc and protein docking, but these are not mentioned in even in your syllabus. If you will able to see the syllabus, not a single word regarding it is mentioned. But I will talk with the authorities, and definitely I will let you know. Don't worry. I will, I will try to help you my level best. No issues at all. Any another software is there? Please let me know. Then I will talk with them also. Any another software is also there? QSAR. Okay. QSAR. Which is there in drug designing. Okay, okay, okay. Yes, please let me know about the name of softwares you want to do. Then I will talk with the concerned authority and let you know. If there is any another software, please let me know so that I will write it down and then I will talk with the team. Okay, structural biology. No issues, just let me know about the softwares which you want to learn. That is only my point of concern, that you will feel happy. Just let me know the softwares which you want to learn. Just let me know those softwares. Even on your own behalf, you can ask them, but I will also put forward your message. No issues at all. Just let me know. No, no, don't be sad. I am there with you. Just let me know, please. Please let me know if anyone has something to say. Then I will, even you can also on your own part, you can talk with them. Otherwise, I will also convey your message. Okay, I had just write the name of the software, which is Autodoc, PyMol, Protein Docking, QSAR, which is drug designing, and structure biology, and virtual screening is also there. So I will just try to convey the message, and you can also do it by your own self also. But it is the only thing which is from my side, which I have to convey to all of you, and I have done almost all the parts which is there. So then, okay, I write down the softwares which you are telling me. We will talk again, and I will let you know, and even you can call or you can ask them by your own behalf also. But that is all from my side, dear. I will share the PPTs with all of you in the group today. Till then, take care of yourself. If you have anything else to say, or you want, if you have any other doubt, please let me know. Yes, please take care of yourself, and I will convey your message. Okay. So till then, happy Diwali to all of you. Goodbye. Take care of yourself and your family. Best wishes from my side for your future, and please stay in touch with me. Okay. Yes, Priya, the extra 10 hours classes are for the review paper basically. So I will convey your message, no issues at all, and then we will see what will happen. Okay, no issues at all. Okay, okay. So my best wishes are with all of you. Take care of yourself. I will definitely put forward your request. No issues at all. But that is the only part which I have to cover, and I have completed it today. But I will put forward your message also. Okay. So that's all from my side. Take care of yourself, all of you, and that is all from my side. So happy Diwali, happy Deepavali to all of you. Best wishes from my side for your future, for everything. Take care of yourself and be happy. This is the most important thing I always told my students, to remain stress free and be happy. And yes, I have one more request to all of you, that if you like my lectures, or as a teacher, if you like me, then please like and share, not share basically, just subscribe my YouTube channel, which is Crash Courses, so that you will be able to get the notification when I will be there on another lecture series. And I welcome all of you to be there when I am delivering any of my lectures. You all are welcome without any payment basically, so you all are welcome there. So just subscribe my YouTube channel also. So take care of yourself. Good night. Okay, bye bye, bye bye to all. Bye bye. Take care. Bye bye. Okay, good night. Good night. Bye bye. Bye bye to all.